So in the last class we had studied on the proteins. Okay, I had given you the introduction. So today we are going to study the protein structure. Okay, the structure of protein. Now I had already mentioned that the protein structures, based on the structure, the proteins, they are classified into four different types of structures: the primary, secondary, tertiary, and quaternary structure. Okay, so here these protein molecules, we know that we can classify protein molecules as monomeric. Remember, monomeric or oligomeric. Monomeric means they have a single polypeptide chain, whereas the oligomeric means more than one polypeptide chain. So more than one polypeptide chain, those are called as oligomeric proteins. Now these proteins, they are further classified based on their structure. Because why? Because proteins they do not necessarily occur in their straight chain. Okay, rather it undergoes a folding so as to take up a three-dimensional shape. Remember, I told you, if the amino acids, when a protein is synthesized, the amino acids are linked together in a polypeptide chain through a peptide bond. But actually the protein structure, they will not occur in this straight chain inside the cells. In order to perform different types of functions, the proteins, they will fold into different shapes. Remember, I have given example last time, such as alpha helix, then we have the beta sheets, okay, then we have the beta bends, and so on. And when these are referred to as the secondary structures. When these secondary structures, they combine, you will see that, suppose here I have alpha helix, now I have suppose a beta sheet, then I have again alpha helix, and then suppose I have a beta bend, and again alpha helix, then beta sheet, like that. Okay, when these structures, they combine together in oligomeric proteins, because we have more than one polypeptide, when these secondary structures, they combine together, they will form a tertiary structure, and so on. We will get another structure, that is quaternary structure. So let us study in detail one by one. Now just remember that a protein, it becomes functional only after folding. Okay, only after folding. So that is why we have different structures described. Now initially, when the protein is formed, is synthesized, that structure is referred to as the primary structure. Okay, so primary structure is simple to understand. The primary structure of a protein thus can be defined as the amino acid sequence in a polypeptide chain. As simple as that. You can define primary structure as the amino acid sequence in a polypeptide chain. So initially, when a protein is formed, it is called primary structure, and at that time it is not yet functional. Just remember this. But when it folds accordingly, it will give rise to the secondary structures, and further folding, it will give rise to the tertiary structure, and likewise to the quaternary structure. So now let us take a look at the primary structure of proteins. Now in primary structure, it simply means the linear chains of amino acids, okay, which are linked together through peptide linkages, through peptide bonds. and at one end we will have, remember, we will have amino group. I have given you the assignments to do this, how these amino acids are joined together. At one end we will have the amino group, and this amino group, it will be free, and the other end it will have the COOH group, the carboxylic group would be free at one end. So in between, we know that these peptide bonds are formed between the carboxylic group of one amino acid and the amino group of the next amino acid. So but the first and the last amino acid, these carboxylic group and amino group will be free. So in a primary structure, there is a specific sequence of amino acids, okay, which are arranged, and it should be a linear specific sequence of amino acids, and the sequences are being decided on the basis of DNA. In your 6th semester, you will study the process of translation, that is how the proteins are synthesized. At that time, you will see, based on the DNA sequence, we know that in a DNA, suppose this is a DNA sequence, suppose it has adenine, guanine, thymine, cytosine, adenine, and so on. Okay, the sequence here. This is the DNA molecule. Then from the DNA, we'll get the mRNA, and then we will, during a process of translation, the proteins will be synthesized based on these sequence on the DNA. Suppose this sequence of DNA, AGT, it codes for alanine. Suppose it codes for alanine, then the first amino acid that will be synthesized will be alanine. Then suppose this next one, 1 codon, that means 3 here, okay? So these three, they will code for, suppose, tyrosine. Then the next amino acid here that will form is tyrosine, and so on. So the sequences of these amino acids here that will form, it is based on the sequence which is present on the DNA. And the cell, okay, inside the cell, when these proteins, they are synthesized, so the cell, it has all the information in the form of DNA, okay, which will determine how these amino acids will be synthesized and how they are going to arrange. For example, like I had given you here, suppose alanine, tyrosine, then glycine, then suppose the next one is serine, tyrosine, and so on. Okay, so in this way, this is a specific sequence which is determined by the cell's information. Okay, suppose this is a one protein. Next we'll have another protein, it will have a different sequence. Suppose glycine, then valine, then serine, then cysteine, and so on. And the next protein synthesis, but we know that proteins, they are very, very large molecules, so not only 4 or 5 amino acids, okay, thousands, a very long chain of amino acid sequence will be formed. But this is just the example to show you that the sequence of amino acids here, they are specific for specific protein, for a single protein. So remember girls, I have given you assignments, for example, like these small peptides. But proteins, they are very, very large peptides. That means you will have a long chain of amino acids, not only this much, okay? So this is just for example for your assignment to do. These are very small peptides. Now, these specific arrangement of these amino acids in a linear sequence, okay, this linear sequence that makes the protein specific for all organisms, okay, for all organisms. Just to remind you, I had mentioned last time also that all are using the same standard 22 amino acids, okay, to make these proteins. So just from these 22, thousands and thousands of different proteins are formed. Okay, so just remember, it is only from these 22 standard amino acids. Now we can see that these these standard amino acids, they are like the you can see they are like alphabets, okay, in English, the 26 alphabets, right? Now you can remember like this, the 26 alphabets in English, we have only 26 alphabets, but from these only 26 alphabets, thousands and thousands of words, of different words are formed. So in the same way you can understand now, it is because a word, suppose like for example here this spelling alphabets, ALPHABETS, the arrangement, this word is pronounced as alphabets. Now if I arrange the words, these words from A to Z in a different way, suppose if I arrange C, A and N, then this word is can. So the same way here, these proteins also, the way these amino acids are arranged, they will form thousands and thousands of proteins, okay? Just like the arrangement, just remember this is just an example, okay, I'm showing, just like the alphabets in English, where different arrangement of alphabets, they will form different words, they will produce different words, right? So same way they will produce different proteins, the arrangement of these amino acids. Now so it depends on how these letters are arranged, different words will be produced. So here also it depends on how these 22 standard amino acids are arranged, different proteins will form. And at this primary structure level, the proteins have become specific for any kind of species, okay, or organisms. But just remember, it is as yet it is not functional, this primary structure, okay. And the peptide bonds, just to remind you, if you remember how the peptide bonds are formed, they are formed by dehydration reaction, okay, by dehydration reaction or by condensation reaction. What happened in a dehydration or condensation reaction? There will be a removal of water molecule when the two amino acids are joined together, okay. So that is how the peptide bonds are formed between these amino acids. And for functionality, that amino acid, this linear chain of amino acid, okay, it has to fold and take up a three-dimensional shape, okay. It has to fold itself and take up a three-dimensional shape. Now that three-dimensional shape will give rise to what we call a secondary structure. When a primary structure is formed, it undergoes folding, which leads to the formation of a next type of structure called the secondary structure. So just remember, secondary structure is formed by the folding of this primary structure, okay. So we will study now how this primary structure, it undergoes folding, okay. And there are three ways for that, okay. Three ways where this linear chain of amino acid, this primary structure, will fold itself in three ways, okay, to give rise to this secondary structure. So how we will define the secondary structure? You can just see that secondary structure of a protein refers to the regular rearrangement of the polypeptide chain, okay. It is the regular rearrangement of the polypeptide chain in space in one dimension, okay. So it is the rearrangement of the polypeptide chain in one dimension. Now the secondary structures, as I had mentioned, that they are made up of three sublevels, okay, or three ways. Now the first one is the alpha helix, okay, the alpha helix. So this structure, it was first described by Linus Pauling. Okay, this structure was first described by Linus Pauling in the year 1951. And this alpha helix, it is the most commonly observed protein secondary structures. So among the secondary structures, this alpha helix is the most common one. Okay, and alpha helix is just like a string. Okay, if you take a look at the alpha helix secondary structure, it's just like a spring, a coiled spring, right-handed coiled spring. Okay, so in an alpha helix, just to show you, okay, wait. Now just to show you the structure of an alpha helix, okay. I will not draw this way, but rather I will just draw this way. Alright, so in an alpha helix, this rigid backbone of a polypeptide is twisted. Okay, it is twisted, as you can see, by an equal amount about each alpha carbon atom. So here, suppose I have one alpha carbon atom, because we know, right, that the protein is made up of amino acids, and amino acids, they have the alpha carbon atom. Remember, one side it is linked to the amino group, the other side it is linked to the carboxylic group. Next, the one side it is linked to the hydrogen and the other side to the side chain. So this is the alpha carbon. So this structure, it is twisted by equal amount about these alpha carbon atom, thus forming a coil-like structures called a helix. So this structure is referred to as a helix, and these are the alpha carbons. Okay, so it is twisted by an equal amount around the alpha helix carbon, and in this helix, the polypeptide backbone, as you can see clearly, they follows the path of a right-handed helical spring, right? It folds this way, so it is right-handed in the right side. A right-handed helical spring to form an arrangement in which each amino acid residue, okay, carbonyl group is linked through a hydrogen bond. Okay, listen carefully. Here in this alpha helix, what happened actually, the carbonyl group of one amino, suppose the first amino acid residue is hydrogen bonded, okay, is linked through a hydrogen bond with the amino group of the next, of the second, or the, of the fourth amino acid residue. Okay, so that means the first one here, suppose this is the first amino acid residue, and this one is the fourth. Suppose here this is the fourth amino acid residue. These two are linked together by a hydrogen bond. Okay, so a hydrogen bond is formed between the first amino acid residue and the fourth, and that it happens throughout the length of the chain. Again here from the first amino acid with the fourth. Okay, so the next, like that it continues. So just remember, in an alpha helix, suppose here if I have the fourth amino acid, okay, and suppose here I have the first amino acid residue, then these two, they will link together in one turn. Okay, in this one full turn, the first is linked to the fourth amino acid through a hydrogen bond. Okay, and in this one turn, okay, in this one turn of amino acid, it contains about 3.6 amino acid residue. Okay, so one full turn of this alpha helix, it consists of 3.6 amino acid residues. And also the distance, okay, the distance between the each amino acid along this helix, okay, it is 1.5 angstrom. So these are some of the important points that you have to remember. Okay, these are the important points, that the first amino acid is linked to the fourth amino acid through a hydrogen bond. Okay, a bond is formed between the carboxyl group of the first and the amino group of the fourth. This is the first point. The second point you have to remember, like per one full turn like this of alpha helix, there are about 3.6 amino acid residues. And the third point you have to remember is that the distance between each amino acid residue in a helix is 1.5 angstrom. Okay, so now coming to the fourth point. The fourth point is that this structure, it repeats itself every 5.4 angstrom along the alpha helix. Okay, it repeats itself after every 5.4 angstrom along the helix, and thus we can say that the alpha helix, it has a pitch of 1.4 angstrom. Okay, so these are some of the important points to remember. And thus the alpha helix, it has a rise pair residue of 1.5 angstrom. Because why? The distance between each amino acid residue is 1.5 angstrom, and the structure, it repeats itself after every 5.4 angstrom. So let me show you this diagram here. Okay, now you can see here, this is the simple structure, and these red in color, it shows the alpha carbon atom of consecutive amino acid residue. Okay, these are the alpha carbon. Now this distance you can see between each amino acid, between this one and this one, is 1.5 angstrom, and in nanometer it is 0.15 nanometer. Okay, it is 0.5 nanometer, 15 nanometer, and this distance between each amino acid residue is also known as a translational distance. And here you can see that pair turn, okay, this structure pair turn, it repeats itself, like this folding, it repeats itself after every 5.4 angstrom. Okay, so per turn from here to here, it is 5.4 angstrom or 0.54 nanometer. So this structure here is the, when you view the alpha helix from the top, okay, when you view from the top, you will see that it looks like this. This alpha helix, actually the spring, I hope you understand, when you take out a spring, when you take a look at the top, you will just see a round, a circle. Okay, but here in this case, what are these structure? You will not only see a round circle like this, a structure, when you look from the top view of the alpha helix, but you will also see these structures. These are nothing but actually the side chains, they are projecting outside like this from the alpha helix. Okay, they are projecting outside. So when you view the alpha helix from the top, okay, suppose a tight spring, if you take a look from the top, you will not only just see the circle, but you can see clearly the side chains. Okay, in this way, they are arranged in this way. And this is the sectional view of the alpha helix, which shows the position of the R chain. Okay, projecting in this way. So an important property, of this alpha helix structure, which applies to other structures as well, okay, is cooperativity. Now that means, for example, here once a single turn, suppose this is a straight chain, right, a straight polypeptide chain. Suppose here's first a single turn, if a single turn starts to form, okay, just a single turn starts to form in an alpha helix, okay, if it has been formed, then addition of these successive residues becomes much more easier, okay, it will becomes much more easier and faster, because this first turn of the helix, it acts as a template upon which to direct these successive helical structures. So just once a single turn has been formed, then the addition of the successive residues becomes much more easier, and that it happens with other secondary structures as well, okay, with other structures. Now another important point to be noted here is that the alpha helix, it can be either right-handed or it can be left-handed also, okay, it's just that this structure, the left-handed helical structure, is not very common. It hardly occurs because it creates a steric hindrance, a steric interference between the side chains or the R group. So it is not favorable. This left-handed helix is not very common. And this right-handed helix is found in almost all proteins. Okay, it is found in all the proteins. Now, for example, the fibrous proteins, okay, fibrous proteins, I have given you example that day, alpha keratin. Okay, alpha keratin is one of the example of fibrous proteins in which a major structural component of this protein is alpha helix. Okay, alpha helix is the major structural component in this alpha keratin, and this proteins is, they are commonly found in the hairs, then in scales, horns, nails, claws, etc. Okay, so these types of alpha keratin proteins, they are the major component of these types of fibrous proteins is alpha helix. And one more thing I want to tell you about this alpha helical structure is that there are some amino acids, certain amino acids, which are more often found in these structures, in alpha helix structures, okay, and rather than other structures. For example, we will also study the beta sheets, the beta bends, the tertiary, the quaternary structure. You will find that some amino acids, they are commonly found, they are more frequently found in these alpha helix rather than in the other types of structures, okay. In particular, I will tell you one amino acid, that is proline. If you remember the structure of proline, this proline is very much found in alpha helical region. Why? Because it cannot form the correct pattern of hydrogen bond. Why? If you remember the proline, the structure of proline, it lacks a hydrogen atom on its nitrogen atom. Okay, there is no hydrogen atom on the nitrogen atom in proline. That's why it cannot form the correct pattern of hydrogen bond. So for this reason, proline is often found at the end of the alpha helix. Okay, so at the end region of the alpha helix, proline is often found where it alters the direction of the polypeptide chain. Now, since it alters the direction, so it will terminate the helix. Okay, because we know that once the first turn is formed, then the next turn will start to follow the third turn, like that. All right, so what happened here at the end, a proline is usually found at the end of the alpha helix. It will alter the direction of the polypeptide chain because it cannot form proper hydrogen bond. Then this alpha helix, it will terminate. Okay, and not only proline, but there are some other amino acids which are commonly found in, frequently found in the alpha helix, but not in other structures. They are found most commonly found in alpha helix, but they are less frequently found in the other types of secondary structures, such as the beta sheets and the beta bands. Now, different proteins, they have different amount of polypeptide chain, right? I had given you an example last time. For example, we have hemoglobin. Remember, it has 4 polypeptides, 4 polypeptide chains. Now here in alpha helix, actually different proteins also, they have different amount of polypeptide chain which folded up into alpha helices. For example, a protein myoglobin. Okay, this protein myoglobin, it has about 8 number of polypeptide chain which fold into this form. Okay, this secondary structure, that is alpha helices. So myoglobin, it has 8 number of alpha helices. It consists of 8 alpha helices, the structure of this protein myoglobin. So the beta sheets and the other secondary tertiary structures I will discuss in the next class.